Uh, I'm actually really excited to give this talk in this kind of form because I very often give this sort of talk either in the context of uh, uh, more neuroimaging or psychology-oriented talks. But I think this is going to be a real test of like how these concepts and how these ideas actually can translate to some practical uses, which is really what it's like to be a translational researcher. You really are at the forefront of not just getting out of the armchair and actually developing something that can help people. And so where are we going to start? Where I'd like to start actually is with some very well-known age-related changes in cognitive functions. And so, this is not going to be surprising to anyone. Some of us have probably see some version of this figure in every age-related talk we see. The one from Denise Park also shows all of these lines going down. Essentially the sort of narrative of cognitive decline. Fluid intelligence, working memory, fluid processing, speed, all these kinds of things. We don't need to be reminded that those are things that decline with age, but I do think we need to be reminded sometimes that there are things that stay preserved throughout the 60s, 70s, and 80s. Things like language processing is a very stable cognition. Concepts, knowledge, vocabulary, semantics, being able to reason through conceptual knowledge, however, is not only well preserved, but actually improves generally across the lifespan. And I think that this is, as scientists, maybe sometimes we overplay declines because it's where we can find a strong, significant difference. But I think if we're really thinking about not just understanding how our brains change and adapt as we age, but also thinking about positioning interventions, not for simply addressing a decline, but for supporting a preserved cognition. I think that's a sort of general soapbox position that I want to advocate for. That I think we can use these principles to guide interventions. Okay, so what do I mean by that in the brains? So there's also some very well-known age-related patterns of brain change, right? So one of these we published back when I was in grad school, down the hall from here, the K-PASA paper, right? I will never live this down, but this is the posterior to anterior shift in aging. We generally show reductions in sensory regions and increases in anterior temporal and anterior prefrontal regions. It's a very well-documented phenomenon. It's a very generalized phenomenon. It happens across many different kinds of cognition. There have been some really interesting investigations of why this happens, both from a vascular as well as a demographic level. But I'm going to kind of marry the two of these concepts, or this brain-wide shift towards more anterior processing and the more conceptual processing that we tend to rely on as we age. So what are the cognitive factors that underlie this age-related PASA pattern? But also, how can we manipulate this pattern and promote improvements in cognitive performance in aging and disease, specifically MCI, knowing this kind of knowledge? Okay, so if I'm going to teach a small room full of people about what I mean by concepts, we're going to spend a little bit of time with those sort of groundworks. I'm going to attack it from three different questions. First is sort of, how these conceptual representations change with age. We're just going to build a little bit on this PASA phenomenon. Then we're going to shift a little bit to how cortical interactions. We're going to, obviously sort of start in the hippocampus, but we're going to build towards a more brain-wide representation of concepts in the brain. And then we're actually going to get to the TMS stuff, right? So we're going to talk about how we can manipulate these representations of information. And I'll try to lead you by the hand with every analytical tool I'll use for this. Stop me at any point. So let's attack the first question first. How do conceptual representations? So let me talk about what I mean when I say a conceptual organization. So you can think about any particular concept or object in your life kind of as a network of stuff, right? A network of features. So a panda bear might decompose down to a set of features like eats bamboo, it has fur, nose, eyes, et cetera, whereas a knife might decompose to a different set of features, has a blade, has metal, has a handle. There's some interesting analysis we can do about these kinds of features and our emphasis on not just the individual features, but how the features kind of go together as we age. But suffice it to say that you can take a memory and decompose it into its constituent features, and the degree to which a couple of different things show that an overlap in those features, it might be something that you can use later to do a different kind of analysis. What do I mean by that? So we build these really big object databases. Lots of different pictures, lots of different categories. We get people to give us what these features look like. And then what we do is we create something we call a concept frequency matrix, right? Really just a list of concepts and a list of features of that matrix. You just kind of ask people how frequently they list these things. So when people see a cat, 21 out of 30 people are going to say it's a living thing. Same for panda. They're going to say it's a mammal. They're going to say the cat's found at home, but probably not the panda, right? And then you might have features that are more unique to a cat, like purr. So what you have here is you're taking conceptual knowledge, right? You're kind of taking these things we speak in very abstract terms, but we're kind of going to give them some real statistics, which we can then use to compare across items. So, for instance, we can assess the similarity between disparate kinds of concepts. So, a panda and a hat are two very different kinds of concepts with not a lot of conceptual similarity. But depending on what kind of colors they share, a panda and what depending what the hat looks like they might share a lot of visual properties like color, or the edges of that particular color, the sort of visual frequency of an image. So you can see now that we're very sort of slyly decomposing things into their conceptual structure, but also a visual structure, right? Because we want to know what's happening in the brain and how those visual elements translate to a conceptual structure. Okay, so I'll skip that. So what do I mean when I say representation, right? So we spent a little bit of time with the conceptual, with the psychological model. Now let me tell you what I mean when I talk about brain stuff, right? So there's a very popular set of analyses that have really just been taking off in the past 10 years called representational similarity analysis. And what this means is its focus is not just on the amount of activity in a brain region, but the pattern of that activity. So in other words, you might have a particular pattern for a panda and a pattern for a hat in the same brain region. They might activate differently. Some of these voxels are lighter, some of them are darker. But just imagine those little, brain regions, turn them into vectors and correlate them together. So if two things are similar in a certain way, we might expect them to activate in a similar way. But as you might kind of anticipate, that's going to depend on the kind of brain region you're in. If you're in visual cortex, if they look the same, they're going to activate the same. But you might be in regions that don't code for vision, regions that code for whether or not a thing is living or non-living or makes the same sound. That's going to determine the similarity. And we're what are we, 5 minutes into the talk? If you're still with me and you're still thinking about how that similarity might matter between brain and a psychological construct, you are thinking theoretically. You are trying to apply a model of the world to a model of the brain, right? And that's all that we're really doing when we do these kinds of analyses. We take a couple of these models. It's based on the similarity of the objects, but that's really just the constituent property, right? And that's how we can test kind of these alternative models about where conceptual information is in the brain. So it's no longer just about, a panda activated or did not activate a brain region. It's what aspect of the panda, of that picture of a panda, activated your brain or caused a specific response. Okay. So it's subtle. So we can, we can test a couple of alternative models. We can see how the visual similarity predicts the brain similarity. Alternatively, the semantic similarity might predict brain similarity. It's going to depend on the region, right? So some brain regions are going to code more for the visual similarity between things, and some of them will code more for the semantics. You'll see this semantic visual kind of push and pull motif throughout the talk. Okay, so how can we bring all this like cool conceptual stuff into concerns about aging? How can we bring it back to this sort of idea of our conceptual memory staying strong? What evidence do we have in the brain that conceptual memory is getting stronger as we age? And then how can we use it, right? So we did a task in the scanner, very simple object encoding. People are just looking at individual items. They might see the picture and think to themselves, okay, that is a panda. Those of you that might study language know that that's kind of a natural, automatic thing to think of the name of something when you see it. Later on, we bring them back and we ask them, we give them a list of things that they saw and things that they didn't. So very typical object recognition test. They just indicate 1, 2, or 3 if they saw it or if they didn't, right? Older adults, as you might expect, are not as good at younger adults. We actually kind of give a vividness rating while they do this naming. You generally have better memory for when things are more vivid, but obviously there's an age-related decline in that overall accuracy. That's okay, right? We'll not focus on that. What we care about really is what the representation in the brain looks like. So if I draw your attention just to the first part of the slide up here, what we're looking at is the representation for the visual model, so that visual similarity. And when things were more visually similar, younger adults showed very strong representational strength in these primary visual regions. Older adults also show it, same kind of visual similarity effect, but just a little bit less strong, right? So this is, suggests a sort of change in that, or decline in that processing in visual cortex for the visual model, right? In the semantic model, we actually see a kind of shift. So younger adults are processing this semantic information still kind of in these posterior regions, but older adults tend to shift forwards. So they tend to use more anterior temporal and anterior frontal regions. So for instance, in the anterior temporal lobe, there's representational strength. The degree to which that brain model matched the semantic model increased in the ATL, and it was much stronger in older adults than it was in younger adults, right? So, this led us to think that basically, there's 2 sides of a coin, right? The effect up top here is well known in the aging literature. So since 2004, the so-called dedifferentiation account has been, this model of how as we age, we form less distinct representations of items. Whereas, the newer finding that we came out with in 2021 and replicated now a couple of times, other labs have also replicated it within the past 2 years, we call hyperdifferentiation. So in other words, we have more distinct conceptual representation in older age than we do in younger age. It's this shift. It's a change from the perceptual properties to the semantic properties of image. And I think it's, there's corollaries to this across both the neurophysiological, the neuroimaging, and especially the behavioral fields, that older adults do tend to rely less on specific details, but more on gist-based or more conceptual knowledge than younger adults. And even in fact, when we look at the strength of this representational strength, the size of these colors that you see here, it actually predicts the accuracy on that test that I showed you significantly in the old, but not in the young. So this is the semantic representational strength. It actually does support memory in older adults and not in the young. So, interim summary kind of slide. So obviously there's a lot of these anterior shifts in activity. They're very widely reported. When we brought it together in 2008, we were certainly not the first, but we're definitely not the last. We kind of see those shift in activation really as a shift in process, whereas the more recent stuff, I think I really see it as a shift in the information processing that happens in the brain. So it's a different kind of cortical conversation that we have in aging. So let's focus now on the second question, right? So I want to kind of pull you from the hippocampus to the cortex and make some interesting links there. These conceptual relationships have kind of been considered independent. In other words, we focus just now on the anterior temporal cortex. There are other regions we might associate with different functions, but it's no surprise to say that we're going to be doing these kinds of representations in different ways with different brain regions, right? So for instance, if we are given a set of images, again, let's think about those same images. The perceptual memory processing might happen if we think about the visual properties of that particular stimulus, the color or the shape. And it might be useful for when we are doing a perceptual task, right? Whereas if we think about the conceptual properties of that particular stimulus, that it was pasta, that it's foods, it boils, etc., that's going to be useful for what we would call our conceptual memory, right? And so, we kind of want to just put our money where our mouth is here and really say, if we believe that these forms of processing this is what's happening in encoding, then these actually should be what's important for later retrieval and not the other way around, right? So conceptual information shouldn't necessarily contribute to perceptual. So we made a big model that does this, right? So we brought together these sort of hippocampal cortical interaction analyses. First off, we'll include things in the model just like activation. So that's the red here. Red is pattern similarity. I'll skip over that for now, but the representation in, let's say, hippocampus or in some cortical region here, visual cortex, independently could predict perceptual memory, or we might actually allow regions like visual cortex or angular gyrus to interact with some of the hippocampal properties that we know are associated with memory. The activation of a hippocampus, so that's this red color again here, sorry. The pattern similarity of hippocampus, or actually whether or not the hippocampus is coding for this visual or conceptual information itself, right? This is sort of a way of thinking about, I wouldn't quite call it informational connectivity, but it's kind of a sort, right? It's kind of asking whether or not if the hippocampus and another cortical region are both engaged in the same kind of processing, is that actually what's going to support memory rather than all these other things, right? So that's the real target of this. And I think when we think about this, let's think about how the hippocampus might interact with these cortical regions. You could have a different kind of style of all of them. So if you have a cortical representation and a hippocampal representation, right? They could be additive. In other words, that when you have more cortical and more hippocampal, you kind of both lead towards this increase in memory. So the y-axis of everything I'm showing you here is the subsequent memory effects, or just in general, just you'd see it as a memory effect, better memory. Whereas on the other two axes along the bottom here, cortical representation and hippocampal representation. So if it's additive, it means that everybody's adding to the overall contribution. There's no nonlinear effects here. It's a completely linear addition situation. You could have a sort of hippocampal gating. So in other words, your cortical representation, the more your cortex thinks about those conceptual features, it's not going to matter, right? You need the hippocampus in order to have a strong formation of memory. So it doesn't matter until you get sort of the back parts of this 3D graph, and then you'll get these improvements in memory. Only when the hippocampal involvement is higher, right? So that we will call hippocampal gating, because the hippocampus needs to make its contribution before we can move forward. There's another kind of, and I think this is maybe the more interesting one to think about, which is a sort of hippocampal cortical coordination. So in other words, it's not just when the two of these are working together, but it's really anytime they're in sync. So anytime the cortex is doing what the hippocampus is doing, you're going to get good memory. One side of this graph is easy to think about, right? So just the additive function of the hippocampus coding for information, the cortex is coding for information. Great. All the information is there that we have, right? But we also have to think about when we're the other side, at the other corner of that graph, it's actually important when we have extraneous information that our brain regions are in sync with not thinking about that. If the cortex is thinking about something and the hippocampus is not, that kind of breaks down our idea that cortical hippocampal syncing is important, right? So you should also have good memories, better subsequent memory effects when those two brain regions are not coactive or coding in the same way. This is essentially the pattern we found. So what we found is that when perceptual memory is predicted by the interaction of hippocampal activation, we had a sort of hippocampal gating effect here. So when the hippocampus was activating more and the cortex in occipital region was coding for visual information, the visual properties of this pasta, that's when we got subsequent memory effects, right? A pretty strong gating effect. Whereas in conceptual memory, we had a different, a similar pattern for these inferior frontal gyrus. So instead, it was actually the coding of this semantic information in inferior frontal gyrus. So we had the sort of same kind of gating mechanism. Also, hippocampal activation was the gate there. And we had these nice interactions. It really kind of shows how you can think about two regions. Again, I don't want to say this is connectivity because it's a little more complicated, right? It's about a deep relationship between two regions instead of simply, a correlation of their timescales or something like that we might do for connectivity. We did not find the effects in the cross comparisons. So, we find essentially what we're calling transfer appropriate cortical representations and how they're supported by the interaction with the hippocampus. So we're going to make a conceptual link from the result that we see there, right? That we have to have this integration with regions in the hippocampus and cortical regions to actually use something that I've been doing for about 5 or 6 years now, which is transcranial magnetic stimulation, to see how we can actually affect these representations more directly. So for those of you not familiar, this is a depiction of a participant receiving TMS. It's essentially a magnet that has a current pass through. That current creates a magnetic field around the cortex. And then when you put a magnetic field near an electrically sensitive medium, you create an electric field. And we create electric field in the brain. It is on the order of maybe about half a Tesla to a Tesla and a half, depending on where you're at. But it can be used to create depolarization of axons and cause neurons to fire. This is a depiction of our Brain Stimulation Research Center, which I co-direct in the Psychiatry department. We usually bring a subject in. We've actually been able to engineer a robot that keeps the coil in the same place if somebody fidgets around. A lot of the studies at the BSRC are actually interested in psychiatric questions. So TMS is FDA approved for depression, OCD, a number of different psychiatric disorders, PTSD. But there's nothing especially psychiatric about the device, right? It just simply has been applied in those situations. We use it to affect brain activity, and not just brain activity at a particular site. We're not just simply hunting around for a specific locus to stimulate, but actually we're trying to affect the whole brain and interactions there. Okay. So there are a number of studies that have been performed in the past 10 years or so using TMS in AD-related populations, largely in MCI, I'll say, just because it tends to be a little less feasible as you progress further in disease. But in general, so what you're seeing here is a meta-analysis of a number of these papers. And I guess what I'll point out is that the sort of overall summary statistic of these papers is off of zero, right? The red diamond is not significantly touching zero, which means that that does seem to be a significant positive effect here. However, it's a moderate effect size across studies, but every one of these studies, used a different outcome. So some of these studies were using change in MoCA, some of them were using RAV-LT, some of them were using a bespoke memory test of their own. So I think the field is going to take a while before it really converges on what an effective treatment might be. If I'm in a more clinical audience, I usually try to scare people and say that I kind of think that there's going to be an FDA-approved treatment for AD using TMS, I don't know, within like 7 or 8 years. And that sounds really early to a lot of neurology and even psychiatry folks. But these studies are currently at phase 2 and 3 in the process. And we have to remember that FDA approval happens because of efficacy, not because of understanding, right? So we have a lot of knowledge about how some of these studies might happen, but I would say that in general, what they're doing is applying what has worked for depression and applying it in the same brain regions, using the same frequencies, and really kind of, not looking necessarily at the same outcomes, but using that same kind of format of just trying to find change in a MoCA score as evidence that something worked, right? So, clearly the first half of the talk is going to matter for the rest of the talk. We're going to talk about brain mechanisms that actually might be mediating this effect here. Okay. So their target is generally left DLPFC for a lot of these studies, but there is this small growing set of studies that is more cognitive neuroscience informed. It started with a young adult study in 2014, published in Science from Joel Voss's lab. The logic they had, so if you look first on this left side of the picture, the logic was that they wanted to affect the hippocampus, but they wanted to use TMS. And TMS does not have good penetration. It gets down maybe about 2 to 3 centimeters into the skull, which means you're not going to affect deep brain structures like the hippocampus directly. But what Joel's lab did is take a resting state scan and seed in the hippocampus and see what kind of cortical regions show a correlation there. What kind of functional network do you have there? And pretty consistently, there was correlation in medial parietal, precuneus, and lateral parietal cortex. Usually a little MPFC as well. Those of us in the neuroimaging field know that this is like probably default, but we can come back to that. Subjects then enjoyed about 3 to 5 days of TMS and then came back afterwards to do a face name association task. And the stimulation group showed improvement compared to sham, right? So, this lab repeated the same kind of paradigm with variations on the theme. It's a really strong body of work and very consistent, showing these improvements here. This is D prime, again, in a face word association task, using this hippocampal network. Another use of the hippocampal seed to pull out these sort of lateral parietal targets, which you can access with TMS. And then trying, different forms of TMS, continuous versus intermittent theta burst. This is from Molly Hermler, who's now down at FSU, was a postdoc in Joel's lab. There's actually Yinghua Chen, who actually was at VX. So I don't know if anybody remembers Nan Kuei and Yinghua. They were wonderful people, and I was sad to see them go. But Yinghua has done an amazing job at Arizona, investigating a lot of these same questions. Using TMS and neuroimaging, TMS guided by neuroimaging. She used a very similar kind of face name association task. And in a cohort of just 9 MCIs, they were able to find these really nice within subject effects of improvements in memory relative to that subject's sham with intermittent theta burst and reductions in performance with continuous data burst. The ITBS, CTBS thing is kind of in the field been taken to mean a sort of excitatory and inhibitory. I'm not a huge fan of that relatively simplistic designation, but the point is they tried a couple different kinds of TMS and they affected things. The way they got to they chose their stimulation site. It was still a parietal site, but instead of doing this functional targeting, they actually used, they pulled out the hippocampus and they ran a bunch of DTI, and they used the structural terminus of a pathway from the hippocampal cortex to someplace in inferior parietal cortex. That was how they chose the site, with the idea that, you know, you're kind of accessing the hippocampus at a distance that way, right? Really kind of fun little study. So, you know, let's talk, you know, I've talked about why it's an effective target. Let's talk about why it would be a good target anyway, right? So why, not just in terms of where it's linked to, in terms of hippocampus, but what does it do? So this angular gyrus, this inferior parietal angular gyrus region, it does semantics, really. I think if you go across the cognitive neuroscience literature, the left angular gyrus is very often associated with either attention or semantics or some interaction of the two. So it's been seen by Jeff Binder for over almost 15 years now that as a sort of semantic hub, a region where lots of abstract representations kind of coalesce into one region. This has been done even with studies not focused on memory per se, but just looking at the overlap of this representation of words and pictures. What's common between words and pictures, right? The way that a word looks is very arbitrary. It's just a set of black and white symbols on the screen, right? But if they have any kind of overlap, then it suggests in the brain that that's where that conceptual kind of representation, that kind of amodal representation might exist. And that tends to be, again, very much in these sort of inferior parietal regions. Other studies have found this. You know, I can show a host of them, but let's kind of move through this. Lots of studies that show that the angular gyrus is a semantically focused region. Okay, so what have I kind of done for you in the past 10 minutes? I've shown you these different accounts for why lateral parietal cortex angular gyrus is a good target. And the first slide made a very strong systems level explanation, a sort of network, a hippocampal network idea. Whereas the second slide is trying to focus a bit more on the representation and what that region does, what kind of processing. These are not mutually exclusive arguments, right? This is very much, the elephant metaphor with a bunch of scientists touching an elephant, and somebody's touching the legs and it says it's a tree, and somebody's touching the trunk and he says it's a snake, that kind of thing, you know? So suffice it to say that nobody's really taken this representational angle using TMS as a treatment for AD-related memory changes, right? So here's where we're at. We're going to focus and use this representational analysis to show how the constituents of memory, this content that I've been telling you about this whole talk, how that actually might change with TMS. Okay, first big question is, can we change representations at all? Like, it actually sounds like kind of a tall order to be able to affect what you think about when you think about Panda, right? Your cortical representation, what Panda is to you, right? I hope that sounds interesting. So what we did is we did this study. This was almost 10 years ago now, where we showed people images. We actually looked at sentence structure. We wanted to get at sort of abstract semantics. And we had a sort of identical recombined kind of memory process here. I would show you a picture of the TMS coil in the scanners. We actually did the TMS coil inside the bore of BAC5. I don't have a picture of that because you can't stick cameras or cell phones inside of BAC, but that's sort of what it looked like. And this is the coil holder that sits in the bed of the magnet, and we kind of, sort of awkwardly position the MRI coil around that. And subjects they received offline TMS, so they received TMS and then they did the task, as opposed to receiving TMS while they do the task. We would call that online. And so they received either 1 hertz or 5 hertz TMS. Again, these 1 hertz and 5 hertz, 10 years ago was sort of like an inhibitory excitatory. We think a little bit differently about it now in the field, but that was our reasoning back then. It's not uncommon in the field. And then we did these kinds of representational analyses. So first, if we look at retrieval, just the representation of the semantics in at retrieval, we found that when we TMS'd at 5 hertz, we actually improved the representational strength in these temporal regions for these items. It actually was linked to memory. This effect was correlated with memory. And it actually had a significant difference between different kinds of TMS. So when we did 5 hertz, we improved it. When we did 1 hertz, we actually made it worse, right? We also did a kind of analysis known as encoding retrieval similarity. So this is the idea that if you, when you're presented something and then when you remember it, you think of the same thing. So you bring to mind that representation. And we found this really nice effect in hippocampus, that the specific item was, that ERS was boosted by 5 hertz and depressed by 1 hertz activity. So the answer is yes, we can affect representations in the brain. Okay, so now we're going to shift to a study where we actually used and applied this in MCI. The study design is pretty similar to the things I've been showing you before. We're going to do a little bit more than we've done before. So we have a very similar object encoding task. Clearly these are objects that we know really well. We know the semantics and we know the visual properties of these tasks. And then after they receive TMS, they're going to see these objects in the scanner during encoding. We get them to process them deeply by actually asking them, are they related? So we'll show them sort of pairs of these items. But really, we're interested in the representation of the individual items. We assess the pair stuff later. Afterwards, they come out of the scanner, and we do a conceptual retrieval, show them the words, and ask them, is this a word you saw, or yes or no? And then perceptual retrieval after that. You have to go this way, then this way, because otherwise it's a sort of re-encoding of the object. So the way that we did this is we had first a sort of baseline localizer to find out the region associated with that kind of semantic processing. And that's where we stimulated in parietal cortex, generally in this sort of lateral inferior region, although there's some individualization there. They then come back for 3 days in a row. So Monday, Tuesday, Wednesday of getting theta burst stimulation outside the scanner, and then going in the scanner and doing this task again, and then coming back out and doing the retrieval, and doing that 3 days in a row, right? So 3 days in a row, why that? It's kind of halfway between, what might be a typical course of TMS treatment. Not only for depression you get it about 20 days in a row, 20 to 30 days in a row. But even the studies that I mentioned before that are kind of doing this, hammer and nail approach, applying DLPFC to, applying TMS to DLPFC for about 20 days in a row in a set of AD or MCI participants, then looking at MoCA score afterwards, right? So you can see it's kind of in between that, right? So I don't think my subjects would be amenable for 20 straight days of scanning. That's nothing that neither my budget nor the subjects could tolerate. But it gives us this kind of slow progression of if there are any changes over time, and whether or not there are just any effects of TMS in any case. So we tested 28 MCIs now, so 14 in an active condition, 14 in sham, with a MoCA range of about 17 to 22. Most, I think 90% of these participants came through the neurology clinic, and I'm very appreciative of that. There's a number of them might have had some interaction with ADHD. In general, this similarity function does, the people seeing the things as being related does tend to promote memory. But the really interesting effect that we focused on was do we improve memory? And do we improve it selectively? So we did show that We can improve AD conceptual memory over time. So you can see the dark blue line compared to the light blue dashed line. There is a significant difference after 3 days. Conceptual memory was a bit stronger than perceptual memory just because that test is, it's a little bit harder for the perceptual retrieval. Healthy adults can do this task over the course of a day, but MCIs we had to test like right after scanning. And so perceptual memory is really going to decline like immediately after you see it. If we were to test, working memory like right after they saw the images, perceptual memory might be superior, but it has a big drop off. Conceptual memory is more resilient because it's more abstractable, right? So we get the effect. We improve conceptual memory. And even kind of to the point that we mentioned before, we look at the similarity of the two things. And so if I show you two objects that are pretty similar related, you're going to endorse their similarity and you're going to be able to form a more coherent kind of pair representation of those. And so this effect, this change in D prime was actually higher for the high similarity compared to the low similarity trials. So this is a nice demonstration of like, what actually are we affecting? What are we selectively targeting? We chose a conceptual memory target and we're affecting conceptual memory, okay? And so the next two slides are actually just going to be what's the brain correlate of that effect, right? So we looked at these semantic representations. I'm coloring everything from 0 to about -0.2. That's again, the correlation between this brain similarity pattern and the semantic similarity pattern, right? Kind of like a second order correlation, a correlation of correlation, right? And really, I just want you to focus on, you can see here that the big differences between pre and post stimulation after the 3 days was that you had these very localized increases in semantic representation in angular gyrus, right? In a couple angular gyrus regions. Those were present in the active. They were not present in the sham condition, which I'm not showing you here. But actually, what was really cool, this was really sealed the deal for us, was that the change in that brain effect, that representational strength, was correlated quite strongly with the change in conceptual memory strength, right? So you had this really nice cortical correlate of the improvement that we engender with TMS. So semantic representations, they increase that parietal, after that parietal TMS, and they're associated with conceptual memory. We did not see the same thing with perceptual memory and the visual representation. So we were not looking for this. We were able to increase the representational strength in visual cortex. Strangely, it actually decreased the amount of visual representations in angular gyrus. Maybe this was led to the, I think people, when MCIs see 120 images to remember 3 days in a row, I think what we were dealing with was a lot of visual interference here. So it's a bit of a challenging task. I might have given them a day off in between if we had to do this again. But, something this does show is the selectivity of what we did. We found changes that were selective to conceptual processing. So, I just want to kind of come back to this. I'll sort of stop on this general summation slide. There's these different accounts for why this might be a good target for future interventions for AD-related disorders. So, the systems level approach of activating a hippocampal network has now been pretty well worked out on both a functional and structural basis. But we're trying to add on to the representational account of that. But that's not the only account of why this might be a good brain region to target. There's also, these lateral parietal regions are one of the earliest regions to show tau accumulation outside of MTL. And this is an effect that we chase quite a lot because it might lead to how these regions are functionally connected offline. There's probably an attention argument here. There's a whole literature, cognitive neuroscience literature, about the interaction of memory and attention in parietal cortex. That might be what we're affecting. Again, there's these age-related shifts we talked about before, whether or not the kinds of activations we're seeing are related to the kinds of representational changes we might see before. And maybe these parietal regions work because older adults are shifting towards a more abstract parietal-based representation of everything, right? Of their, at least with respect to our object stimuli, but maybe even more towards more naturalistic stuff. Yeah, so that's just sort of summary. These are kind of little icons of the stuff that I showed you. These the first question was focused on arguing to you that we kind of shift from these posterior perceptual representations to more anterior conceptual relationships. That the hippocampus interacts with these cortical regions in a sort of gating fashion, and really that this kind of understanding sort of leads really to what the kind of thing Heather was saying before about how like understanding these patterns might actually help you improve the outcomes in older adults and understand why they happen, not just that they happen. So I'll just thank my lab. A lot of great work here from postdoc Paul Bogdan, grad student Matt Slayton, and Shen Yang. Mags and Miriam were stellar CRCs. I miss them a lot. Mags is over at UNC now in speech and language. And obviously my collaborators, so Roberto's a strong lab. We co-direct the lab together. And Uncle Peter Cheff has been absolutely instrumental for making all these crazy TMS paradigms work. I'll stop there and see if there's any questions.
Thank you so much. That was fantastic. Really appreciate just a really incredible amount of work. And I think, I don't think I lost you too many times there, so that was good. Yeah, I think delivered really, really expertly. Too, but I'll open it up for others first, and if they want to take a minute to think about deep burning questions, I can start. So just go ahead and raise your hand or speak up if you'd like. So I guess I was sort of thinking about at what stage in disease you think TMS might be more or less effective, and if there's sort of like therapeutic window that where the sort of biology of this makes the most sense, and is that just sort of like, well, as early as possible, or is it potentially a little more nuanced than that?
Yeah, I mean, that's our default, right? Working as aging researchers,
Mhm.
always, how can we attack it before it's too late, quote unquote, right? Um, and what I think I thought a lot in that mindset, especially when I was recruiting, especially when I was trying to really just make the studies happen. It's always about, what kind of how late can we make the ages? How well can people do the task? What I generally found was it really didn't matter. There's this question about compensation in the field, right? Who are the individuals that compensates? Are they the individuals that are most that need it the most? So the individuals that are performing the worst, or is it the individuals that actually have the reserve to find compensation, right? And I think once you start individualizing treatments like we've done in the past study, it doesn't matter. They will sequester into different kinds of groups, and they might use different kinds of brain regions to do what they do. But so long as you're attacking functions that remain relatively preserved across the lifespan and afford individuals a different means uh, of finding that compensatory pattern. What I mean by that, it might actually be a different cortical pattern or a different cortical region for compensation. Then you can start to apply this at any part of the MoCA scale or the performance scale, right? I think one thing that I've learned about this study is that when we shifted from sort of, random effects across subject kind of investigations to within subject comparisons, using these kind of multi-day data that we have a lot of brain data. I mean, they have 4 different scan sessions. We're able to find like fairly replicable, fairly consistent patterns of individuals using. Some of them used the parietal representations, as you saw in the correlation. There were subsets, however, that had a stronger representation of this information in frontal cortex, and that was correlated with performance. And significance was not an issue because we have hundreds of trials in each individual and across these days, and it's replicable across days. So yeah, I think in general, like when you're planning the study early on, certainly worrying about the level of cognitive function is important. But I think so long as we're dealing with, I mean, practically, I'll say probably like CDR1 or below is probably who's going to be a target for these kinds of interventions. I think beyond that, they might not be appropriate. But I don't think we're in the ballpark of like you've got to get TMS when you're 45, or this isn't going to work, right? Like I think it is very much about using existing structures and existing representations and just emphasizing.
And then maybe just a quick corollary to that. Do you think that there are potential applications for this in, I don't know, outside of kind of age-related cognitive decline in the, maybe like earlier, more syndromic forms, or, sort of too early to tell? Or maybe there's more, cognitive reserve in those individuals and they're already kind of leveraging that to the extent they can.
Yeah, it's a good question. So maybe like an early onset.
Yeah, like someone with like a really like PARC mutation, or I hesitate to think, I sort of work some in the Down syndrome AD space, right? But there, I think it could be sort of just confounded in a lot of ways by cognitive impairment, more generally, right?
No, I mean there's a whole other kind of literature on using TMS as a performance enhancement kind of thing, right? Less focused on obviating some kind of cognitive decline, but actually on targeting specific kinds of representations. And imagine doing this kind of study that we did, but instead of targeting a conceptual region, you target a visual region. So you show selectivity in the kinds of visual performance. There have been those kinds of studies with respect to face and scene performance within the same study. So you can show very selective improvement after TMS. I think there's a lot of cool work about even the timing of this event. So whether or not you time the TMS to specific onsets and to specific electrophysiological responses associated with that cognition. I think there's a lot of potential applications, across the board. Whether or not they're there in other specific syndromes, I think it would be really the goal is to sort of identify what kinds of representations are preserved and what kind of and can be promoted. I think this is very much a wind at your back kind of metaphor. I don't think TMS is never going to allow you to do something new, right? It's going to allow you to do something that you currently do better or more efficient. And so I think zeroing in on whatever that is for the particular syndrome and then focusing on promoting it is probably good.
So Bradley Cooper and Limitless, right?
Not outside the conversation.
All right. Anyone else that wanted to ask a question, or Jenna perhaps, or anyone else? Check the chat too.
Sorry, any ETI in here. I have a data set. I think that Tyler is probably going to attack it at some point of seeing if TMS connectivity from parietal cortex changes over these 3 days. We have a Muse scan every day that they do imaging. So
that's awesome. Yeah, that seems like good data to address that question, and I think Tyler will be a good person for it.
Yeah.
Just one question that I had during this was kind of like it's more of a cognitive neuroscience question, but like what is the exact role of angular gyrus, like mechanistically? Like, do you think it's more of a promoting encoding sort of mechanism through the TMS? Or do you think it's all like the TMS has long-term effects for the later retrieval stage as well? Or kind of like what is its role exactly in improving that memory score?
Yeah, so uh, the angular gyrus really enjoyed a big spotlight about 5 or 10 years ago. So there was a model that Roberto developed called the ATOM model, or the attention to memory model. And what it really focused on was sort of dorsal versus ventral parietal cortex. Um, and you know, the ventral cortex being more associated with attention to internal forms of memory, or sort of engrams per se, whereas to more dorsal attention is a little bit more of like a Dave kind of thing of focusing on general awareness, general attention to exterior elements of memory. So the role of the angular gyrus in the tasks that we've done, we have to sort of assume it's an encoding process if we're finding any effects of TMS, because the effects of this TMS probably dissipate after about an hour, and by that time, they start retrieval about an hour and a half into the whole process. So what I think is happening here is that they're benefiting from the ability to sort of see these exterior representations, but actually encode them and get them into an MTL system, and turn them into more internal representations, and focus more that way. Are they benefiting at retrieval? So at retrieval, that's when you really need that attention to internal memory, right? You've shown the word panda bear, and you need to access that internal memory. It could be that there was sort of lasting effects at retrieval for that angular gyrus representation. We looked at individual differences and whether it was more dorsal or ventral stimulation. We didn't really see anything convincing there. But that would be my sort of guess on what's happening in angular gyrus.
Super interesting and great talk. Good to see all the work put together.